Hello again. At some point you'll notice that I tend to have to break the videos into shorter segments. This is for two purposes. I want to be able for you to actually read shorter or watch shorter videos, but also to keep the files small. So you know. Right, so we stopped here in the previous video, so I'm going to backtrack a bit and look at this table 15.2, where it says the consequence of point mutations on polypeptide structure and gene interaction. Again, it's divided the consequence based on site. You have the coding sequence. Down here you have the intragenic non-coding sequence, and then you have the extragenic. So within the coding sequence, it depends on whether you have a silent. So you're talking only about point mutations in this table. So whether it's synonymous, whether it's missense, whether it's nonsense, whether it's read-through, whether it's frame shift, whether it's non-frame shifting indel. I explained in class what indels are. And then when we talk about in intragenic non-coding, so it can be within the introns and untranslated region. You would have thought that if you get mutations in introns, it'll be okay, but not necessarily, because sometimes introns have regulatory elements. For example, here, mutation disrupts an intronic regulatory element such as an enhancer. An enhancer is a particular sequence in a gene that is bound by other proteins that will make it express even more. So if that region, normally this enhancer is located within the non-coding sequences. Or if that region is mutated, so that means you don't get the proper expression of that particular gene. In intronic sequence, the mutation abolishes the splice site. Splicing of exons requires certain sequences to be recognized by proteins, so that sequence can be mutated, and normally the sequences are not within the coding sequence, is in the intronic sequence. So, or you have a mutations that create or looks like a new splice site, so that it confuses the splicing system. So mutations, if you're looking for mutations, causative mutations for particular diseases, when you find mutations in intronic sequence, you cannot just dismiss them. You have to really look at all the possibilities that it could actually be the cause for mutation for that disease. So you're not just looking for mutations in the coding sequence. And then of course, extragenic means coding sequence that are outside the genes, so you do get mutations there. Is it important? Yes, if it hits a regulatory element. Again, gene sequence are the coding sequence, and then gene-related sequences are the regulatory elements, and these regulatory elements will be present in the non-coding sequence. And so now more and more sequences are now being identified as regulatory elements. So yeah. So I should actually time myself. So okay. So the next table, 15.3, consequence of macromutations. The previous one was consequence of point mutations. So this one is the consequences of macro mutations and chromosome imbalance. So again, what effects are you expecting? So if you have a whole gene deletion, of course, you abolish gene function. And this is where the term haploinsufficiency, haplo means half, insufficient is not enough, may occur as part of larger gene deletion, resulting in a contiguous gene syndrome. So that means multiple genes are being lost. Partial deletion depends on the extent of deletion. You may just delete, you may experience just a small segment. That is, you know how genes fall into proteins, right? So you may lose a certain region of the gene that is not important. You may lose the region of the protein that is not important. So the gene may still function. But if you are going to lose like half of it, the protein, so that's probably will probably result in a more severe effect. So partial deletion, truncation, or internal. So truncation is losing at the end. Internal is something you lose in the middle. So it depends on the extent of deletion. Deletion within introns are often neutral unless a regulatory element is modified. Right. So again, if you don't understand, read it slowly. Partial. This is duplication. That means you increase in dose. So when you get duplication, normally you're talking about the dose, gene dosage. Why? Here it says the products are titrated against the product of other genes. Sometimes the amount of the gene determines whether it functions or not. Yeah. So the amount of one gene determines the amount of other genes. So this is where, you know, this kind of mutations will play a significant role. So if you have disruption of the deletion of rearrangement, loss of function, and then chromosome loss or gain, and so on. All right. So I think these sections continue to explain the table. So this is a functional consequence of point mutation, functional consequence of macro mutation. Macro mutations, one of this one I've highlighted here, gene diffusion effect. Sometimes two genes, you know, when you have translocation, sometimes two genes fuse, and you do get hybrid genes, and some of them have been known to cause cancer. For example, the BCX, ABL, BCL2, I can't remember, but there are examples where a fusion of a gene actually results in a new kind of protein that is deleterious. So again, so they maintain the commutative balance with product of other genes. This is have been mentioned before. Mutations involving repetitive DNA. When you talk about repetitive DNA, the problem is always when they want to, you know how chromosomes, homologous chromosomes will pair, and in a replication or in when you do crossing over, they will pair based on sequences, but because it's repetitive, they may pair wrongly. Look at this examples, you know, because these blocks are similar sequences, so they may result in unequal crossing over, or they can also result in slip strand mispairing. That means that region has been synthesized as such, but then there is then what happens in this? For example, look at this picture. You will see that, you know, this TAG has moved one unit backwards, and that happens because it slips, because there is also quite stable, yeah? And this is what we call a slip strand mispairing, and this has, both of these are the two mechanisms that result in changes in the length of repeated sequence, and some has been known to actually cause diseases. Alright, before we go further, I will stop here and then I'll join you again in the next video. See you later.